Welcome. It's an honor for me to get to discuss with Dame Sarah Gilbert, Sarah Catherine Gilbert, officially in her new title, her honor of being inducted into the International Society of Vaccines Fellow Society, which is a distinguished group of very accomplished scientists. Sarah, as you know, you were honored for your many contributions to development of vaccines for emerging infectious diseases, including really, you've had a focus on diseases for globally disadvantaged populations. And you've made extraordinary contributions to the development of the chimp adenovirus vaccine for SARS-CoV-2, ChAdOx1, and COVID-19. In fact, that vaccine is the most widely used globally to protect populations. And so we really thank you for your accomplishment. And your career has been a beacon for young scientists to emulate and to think about as they develop their own careers. So I did want to talk a little bit with you about the start of your career and how you chose science. And then we'll go into some finer details if we have time about different targets you've worked on and some of the challenges in research that you faced. And we can hopefully talk a little bit about the pandemic race. So I know that you attended, your father was a shoemaker and your mother was a primary school teacher, and you were born in Kettering, North Hampshire. So can you tell us a little bit about growing up in Kettering, North Hampshire? For you have a sort of wide audience of young scientists who may hear some of this, so that would be useful, if that's okay.
Actually, it was Northamptonshire.
Northamptonshire. I'm sorry, I'm from Brooklyn, so who knows how I say anything. So I apologize for being from Brooklyn.
Well, there is a North Hampshire, but it's not the same place, so that's fine. Yeah, my father was an office manager, mother teacher, as you said. I just had a very quiet, normal life. I lived in Kettering all of my young life, didn't move around. Was there the whole time, going to school there. We had a school at secondary level so from 11 years onwards, it was an all-girls school. I didn't have any choice in that. That's just the system that we had. And so we had all teaching in all the sciences as well as the humanities. And I found, as I went through school, that I found biology in particular really interesting. I was just very interested in how things work, how living organisms work. Backed up by what we were learning in chemistry as well, but just how biological systems build themselves, what they do, what happens when they go wrong sometimes. And so I didn't really know what I wanted to do in the long term, except that I knew I wanted to know more about biological systems. And so that led me to apply to the University of East Anglia to do a degree in biological sciences. It was a very broad degree. And I think that was a really good choice to make at that age, because I think it's a mistake to study a course that's really very specialized from the age of 18 onwards, because when you're applying for the course, it's probably over a year before you start it. You haven't finished even your school training in the science subjects. You don't really know what you might be missing or what you're going to enjoy most once you get into the details of it. So my course involved biology, but also chemistry, physics, maths, little tiny bit of computing, which wasn't a really very big thing in 1980, environmental sciences as well, and just kind of trying to, see connections between all the different sciences. And then as I got into the second year, we had to become slightly more specialized, choose from a range of courses. So I was doing things like biochemistry, microbiology, molecular biology. I think we had 6 different courses at that stage. And then for the final year, it was choose one subject to specialize in. And I chose microbiology because by that point, I got really interested in understanding the details of what goes on inside a cell, or when a virus gets inside a cell, and how all that works. and also the fact that biological systems are not completely reliable. So if you have, if you're thinking about maths or physics, you can have things that are true and they're always true. But in biology, you have things that work this way, but actually sometimes a bit of the time they go slightly wrong and something else happens, and that makes it more interesting. So that's where I got into looking at microorganisms and found them fascinating.
So I think, well, actually, I was going to ask you one more thing. So when you were doing computers, I did computers at the same time frame. Those were those punch cards you had to do that you had to carry in a box and load them for doing your computer programs.
Yeah, yeah.
Almost a stone age to think of the way we do things now. So just the way things were. So you specialized in, you then went and did your graduate work at University of Hull and focused on genetics and biochemistry. And I understand that after that, you joined industry for your postdoc. People making career choices, and you wound up in one of the finest academic institutions in the world. So how did you see that at the time, and how were you viewing, how can other people think about the way you viewed your career and your developing at that stage?
Well, I always wanted to use scientific research to make an impact in human health. And I didn't know how that would pan out, but I wanted to understand what's going on with biological systems and then use that understanding to develop something that could be a treatment, or, as it turned out, a vaccine that would improve human health. And moving into industry at that point seemed like a good way to go, because there's a lot of different companies using biological research and microbiological research in order to develop treatments and vaccines. And I changed jobs a couple of times, and for a while I worked in an academic institution which was actually a partnership with biotech, and we had people from companies who came and worked with us in the university. And then after that I worked in a biotech company for 4 years. And in that company I kind of saw both sides of working for biotech within the 4 years that I was there. To begin with, it was expanding. There was lots of money and lots of energy for research, lots of collaborations with academic groups, doing really interesting things, good collaborations within and outside of the company. And then, for reasons which were nothing to do with our company at all, the parent company decided to sell it, and it moved on to a different parent company, and suddenly there wasn't so much interest and so much investment, and we were kind of almost marking time and doing some much less interesting work, I felt, for a bit, and it didn't seem to be improving. So at that point I decided I was going to move somewhere and looked for something to do that was interesting, because I was quite clear at that point I wasn't interested in working 9 to 5. I was interested in working on something that interested me, and then I didn't mind what hours I work if I really was interested in it. It's it's like it just keeps you going. It keeps you awake at night. It keeps you thinking about it all the time. But you can be creative. You can change things. And that was the environment that I wanted to get back to, which had been the environment in the company when I first joined, but that changed. And as I say, it wasn't anything to do with our results. It was just factors completely outside of our control, which felt very frustrating, that a large company that wasn't even really looking at what our small company was doing could have such an effect on us. And I could see that within that particular company at the time, wasn't likely to change any time soon. The difficulty I had then was that if I wanted to go into academia and write a research grant to start my own group, I wasn't allowed by the company's rules to write a grant on the same subject that I've been working for within the company. But then if you don't have any track record in a field, if I was happy to switch fields, but then I had no track record, and it would have been very difficult to be successful in raising grant funding, given that I was trying to do something new. So although I was offered the opportunity to have some space in a university and set up a group, it would then have been up to me to establish my research, which was very difficult to do coming out of a company and not being able to work on the same things I'd worked for in the company. So I decided instead to look for an academic post with an established research group in a university, and that's how I ended up coming to Oxford.
Right. So that was a big change, and an important change, and that was in the laboratory of Adrian Hill, right? If I'm correct. So how did you, so and Adrian and you have been champions together of vaccines over the years, and of course, instrumental. He was knighted as well, I understand, if I'm not getting that incorrect, for his work also on the Oxford vaccine for COVID-2. So could you tell me about that, how you guys sort of started, how you joined that group, and a little bit about some of the projects you worked on? I know you were doing malaria, I believe, at the time.
Yeah, it was malaria for a long time. And I didn't actually join to work on vaccines. I joined for a genetics project. And it was a project on the genetics of interactions between the malaria parasite and the particular host it was in at that time, and how HLA type and particular genes or variants of genes being expressed in the parasite were interacting. We found there was quite a strong interaction in one particular way. And the only way, this was HLA, class 1 HLA molecules, the only way that effect really could have been mediated was because of T-cell responses happening in the liver stage of the malaria infection. So for those people who don't remember their malaria life cycle, we don't always think about it all the time, it's quite complicated. So there's a life cycle that happens inside the mosquito. The mosquito doesn't just carry parasites from one person to another. The mosquito actually gets infected, and there's a part of the life cycle that goes through the mosquito and ends up with lots of the stage that's called sporozoites in the salivary glands of the mosquito. And when the mosquito then bites somebody, they get injected into the bloodstream. And there, if the person has a very high level of antibodies against the sporozoites before they get bitten, then the antibodies can act against the sporozoite. But the sporozoite gets straight inside cells in the liver within about an hour. So there's no time to generate an immune response against the sporozoites once you get bitten. And unless you've got those antibodies already, they can't do anything. But then it goes into the liver, and there it's hidden away from antibodies inside the liver cells. But obviously, malarial proteins are being produced inside the liver cells. They get processed, presented on the surface of the liver cell. And that means that they can be vulnerable to attack by T cells that can recognize those particular epitopes. And that gives us the opportunity to intervene and vaccinate to induce a T cell response against that stage of the life cycle, while there are really very small numbers of parasites inside the body. And if that was successful, then the person who'd been infected would have no illness, because the illness doesn't happen until the parasite then comes out of the liver and starts infecting red blood cells and amplifying. And as the numbers of parasites get larger, then that's when you start to get the symptoms of malaria that we're familiar with. And from there, a mosquito can bite the person and then take up the parasite from their blood and go on to complete the life cycle. So the focus was initially on understanding this genetic interaction. And then, given the fact that from this work and also some other work on HLA associations in malaria, this had to be mediated through a T cell response, and it had to be at that particular stage of the life cycle. Could we vaccinate to give a protective T cell response that would prevent people from becoming ill with malaria if they were bitten by an infected mosquito? So we then started looking at a number of different technologies that could be used to induce T cell responses. I'd done work on molecular biology in the biotech company I've been working for, so I started making plasmids for DNA vaccines. We weren't working on RNA vaccines at that time, but we were working on DNA vaccines. Also looking at peptides, peptides delivered with adjuvants, various different ways, assessing different technologies, all of which were suitable for taking into clinical trials. We weren't interested in some of the things that are sometimes done experimentally, like using complete prawns adjuvant that you can't use in a person. It was all aimed at being able to develop this clinically. And one of the technologies we started working on was viral vectors, replication deficient viral vectors. So they can't multiply, they can't spread through the body after vaccination, which makes them very safe to use, even if a person has a compromised immune system. And we, over the years, made lots of different prototype malaria vaccines and tested them in animal models and had different degrees of success, and started to work on the fact that if you give different types of vaccines, so a DNA vaccine followed by a viral vector, for example, you could actually get much stronger immune responses. And by 1998, we were ready to start clinical trials. And so I was involved in preparing some of the early applications for regulatory and ethical submissions to describe the technology, and then to apply for permission to do those clinical trials. And then I volunteered to take part in the clinical trials and got vaccinated with a DNA and a modified vaccine, Ankara vaccine that I designed. I designed the DNA to encode the epitope string that was inside those vectors, and we started to look at immunizing people and generating T-cell responses with the vaccines we've been working on in the lab.
Yeah, those are all really important accomplishments across the area. One of the really important, among the several important things here, besides the contribution to malaria, the Primeboost approach, of course, you guys were some of the pioneers in that space. And now we're seeing, post-licensure or post-approval of these different SARS-CoV-2 vaccines, that there's a lot of work on basically prime or boosting or combination approaches that are being studied, including by many groups. So, I mean, that work underpinned all of this kind of discussion going forward. So could you talk about how you see that as evolving briefly, or how it sort of directly links that past early work with trying to create a single, this one malaria program into much more diverse field?
Yeah, so one of the things that happened with the malaria program was trying to develop technologies that we could quickly use to make lots of different prototype malaria vaccines. And that led us to thinking that we could also use them to develop lots of different types of vaccines against other diseases. So we call them platform technologies, and this is a complete sea change in how we view vaccine development from how it happened in the 1960s. So Maurice Hilleman, who created a lot of vaccines in the 1960s, most famously probably the mumps vaccine, started by taking the mumps virus itself, actually took a swab from his daughter's throat, cultured it in the lab, worked on it for 2 years to make an attenuated mumps virus, which could then be used as a vaccine. Now, if we do that for every single pathogen, starting from the pathogen itself, it's obviously a slow process, and you have to judge when you've got the vaccine to a point where it would be safe enough to start clinical trials with, and there has to be a lot of care and attention around that. But if we use technologies such as RNA vaccines or replication deficient viral vectors, most of the work is done on the technology itself, not on each individual vaccine. And that means we can do so much more before we even know which disease we want to make a vaccine against. We've got the flexibility just to add in the gene sequence that we need, and then we can develop it very quickly. And we already know how we're going to manufacture it and what dose we're likely to want to use and what the side effects in the few days after vaccination are going to be, because we've seen that all before with the same technology used in different vaccines. So that was one thing that we were really keen on developing. And so the idea of being able to develop multiple vaccines against outbreak pathogens, viruses that we know about that sometimes cause outbreaks, which could then spread, that is really aided by having these platform technologies that allow us to go so much more quickly. And of course, then trying to adapt them even further so that we can go really fast if there's an unknown pathogen, such as SARS-CoV-2 that comes along. But the work on the heterologous Primeboost has been much more relevant than we thought it was going to be. When we're thinking about manufacturing vaccines for a disease that causes outbreaks, such as Ebola, such as Nipah virus, for example, where there are frequent outbreaks, but they're generally very small. If we want a vaccine against those diseases, we really only want to have one technology involved. We don't want it to be complicated by having to use more than one technology, because then if you're making a stockpile, you'd have to make a stockpile of each of the vaccine technologies. If it's a DNA and then an adenovirus, or an adenovirus and then an MVA, you've got to do both of those, and they're different manufacturing methods and probably different storage requirements, which isn't helpful if you're trying to stockpile vaccines. And also you want really rapid onset of immunity if it's an outbreak. So if you can achieve that with one dose, that's desirable. So we've been thinking about just one dose for adenoviral vector vaccines to protect against any of these outbreak pathogens. And we'd shown in preclinical studies, when we vaccinate animal models and then deliberately challenge them, we could get good protection with one dose. As it turns out, coronaviruses are quite difficult to get solid protection against. And that's why we're using 2 doses of the vaccines in almost every case. Janssen's vaccine is licensed as a 1-dose vaccine. There's no reason they can't give a second dose. They just haven't produced the clinical trial data that shows the efficacy of giving a second dose. It would, I'm sure, increase the efficacy if they were to do that, because it's essentially the same technology that we're using, except it's derived from a human adenovirus rather than a simian adenovirus. But then it's come full circle around to looking at heterologous prime boost again, because of the need to vaccinate so many people around the world, shortage of vaccines. We started working on heterologous prime boost trials really to look at the possibility that when somebody came for their second vaccine, the vaccine they had the first time around might not be available in the same clinic. And so what do you do with that person? Do you say, no, sorry, go away, we can't vaccinate you? Or do you vaccinate them with the vaccine that you do have in the clinic? So that was really the reason for starting the work on those. But now there is data coming out to show, not unexpectedly, that we can get some really good, strong immune responses with heterologous prime boost. Again, it's not necessarily a recommended strategy against SARS-CoV-2, but it's certainly one that can be used in certain circumstances. And it's really important to have the trials to establish the safety of doing that, as well as looking at the immune response. We're still not ready there. We're not really there with a correlate of protection for SARS-CoV-2. We don't know exactly what we need to achieve to get solid protection against SARS-CoV-2. So we'll then need to look at the efficacy of different vaccination regimens if there's going to be any recommendation to use them on a wide scale.
So that brings up another question, and we don't have that much more time, so we'll keep, if we keep this one, because then I want to go to a different issue. So as you know, in the discussions now in the US are to give a third shot of, recommend a third shot of the RNA vaccine. So Prime Boost is now Prime Boost Boost, so to speak. And actually the one DNA vaccine that was approved now in India is a 3-shot vaccine, as you may be aware, the Zydus D, of course. And as you mentioned, Johnson & Johnson is doing 2, and who knows. So how do you see, if you can sort of in sort of a very brief, how is your long view of this war against this particular virus for us? I know that's a big thing to handle in a very short time, but I have a lot of faith in you.
Okay. Well, I think different people are going to require boosters to a greater or lesser extent. So starting with the immunocompromised, they really don't make good immune responses. Give them a third dose, it does improve. It's still not great, but it's better than it was after 2 doses. Older people are losing their immune response a bit more quickly, so it was good to think about boosting them. Young, healthy adults' immune responses look like they're being maintained pretty well. And even if we're measuring a drop in antibodies, they've still got the immune memory that can respond when they're infected. So I think it's going to be quite a complicated story of deciding who to boost for the best use of the vaccines and when to boost them. And it's not a case of saying we just need to boost everybody immediately.
Thank you. So I did want to ask about another thing. So I know you have triplets, and and they're all interested in science.
Yes, they are.
So that's an enormous challenge. So could you briefly say how how that's been? It's of course the world's wonderful experience to have children and to help them become our future. So just if you could share a few words, that would be wonderful.
Well, when they were very small, it was quite tough. As parents, we were outnumbered from day one. Most people get a chance to practice on one child and then decide if they're going to have another one. But we had 3 of them right from the very beginning, so we just had to get on with it. And it was obviously really busy with 3 babies. But as they get older, there are some advantages in having triplets because they're interested in, they're at the same life stage at the same time. So we're not trying to cater for activities of children of different ages. They're all interested in the same thing. They got on really well. They still get on really well, and that's great. We never had to try and separate them and find separate things for them to do. So it's really pleasing to me that they're interested in science, and I hope they find their way. They're really obviously at the beginning of their scientific careers now, so they need to work out what it is that they want to do.
Right. So So the other question I had was, which I showed to my daughters. It's a very exciting thing that you received a Barbie doll in your honor. And so can you tell us a little bit about receiving this type of honor?
Well, I was one of 6 women who had 6 women who'd responded to the pandemic in some way. So there were intensive care specialists, nurses, me as a vaccine designer, and Mattel wanted to show girls the role models in different areas of life, and this was the kind of the pandemic special set of 6 Barbie dolls. And I think it's a bit of fun, but it's also important just that girls are aware of the fact that there are women in lots of different roles in life, and it was part of just kind of showing the kind of career that girls could have if they chose to be interested in. And I partnered with an organization called Women in Science and Engineering. And one of the things that they do is on their website, they have something called My Skills, My Life. And you can go and input what you're interested in, what you like to do. And it will give you some information back about a scientific career that might be the right kind of thing for you. So within our vaccine team, we've got lots of diverse roles. You know, we have people who work with the volunteers, with the people all the time. And then we, at the other end, we have people who just work on the data, the statisticians who just sit with a computer and really know what they're doing with the data to analyze everything that's coming out of that. And then in between, we've got people thinking about designing vaccines, manufacturing vaccines, quality control of vaccines, planning the clinical trials, many, many different things. So there's
Hi, Sarah. Welcome back. Sorry, the Zoom cut us off briefly, but that was a really important discussion you were leading us through about that scientists. Well, many people think it's very boring. We're all just pipetting at the bench all day. Scientists can do lots of different things, different jobs. And so, could you just sort of finish that thought that we got cut off on in the recording?
Yeah, it's just for young people thinking about careers. Don't think that all scientists do the same kinds of things. There are lots of different roles. There's a big crossover between science and medicine and healthcare. And some people are clinically qualified, some people aren't. Some people work with data, some people work with people, with patients or trial volunteers. Lots of different things that people do. And we need a very diverse team. We like to have people from different countries, different backgrounds, and they do different things. And we all work together as a big team, and it's the team that's really important.
Right, I think that's a great message, that there's lots to do. And it is it is where people meet science. That's really what vaccine research is. It's people and science and cultures and families and all the rest. So it is a very humanistic science approach. So the other thing about sort of using the comment about it can be boring to be a scientist, which is not the case. I think your recent award, I know you've received many awards as sort of, I did mention you were now Dame titled, and you have many other awards during this year, humanitarian awards, and you were honored last night with the GQ Man of the Year Heroes Award. So as one of the Men of the Year Heroes Awards, could you tell us a little bit about that? It is a great thing when, to me, that women are winning Man of the Year Award. I think it shows you just what we need.
Yeah. So it's obviously it's long established award ceremony. They have awards in different categories, and the last award of the night yesterday was the Heroes Award, which they gave to myself and my colleague, Dr. Kath Green, who heads our biomanufacturing facility. They made the first batch of the vaccine. And together, Kath and I also wrote a book about our work on the vaccine, which we did because we wanted to tell it from our side. We kept hearing that people were concerned because the vaccine had been developed too quickly and that the corners had been cut. And we wanted to explain how that's not the case. And it's quite a long story. So that's why it's a whole book about that aspect of things. We address vaccine hesitancy and we explain what happened in 2020 to get that vaccine developed so quickly. And we were then selected with the team to go to the award ceremony to receive the Heroes Award. So we took 8 other people from the vaccine team, which were randomly selected, They all have different roles in the team, and we went and were honoured with the award and lots of champagne, and then took selfies with all the celebrities that were there.
Well, yeah, I think that's a wonderful thing. Do want to ask you one more question, which I realized. So you worked very closely with AstraZeneca and Tanya Villafana's team. Could you tell us a little bit about that relationship and collaboration? Really championing this vaccine.
Well, obviously, as a university, we were never going to be producing this vaccine for widespread use. That has to be done by a company. We knew we could go a long way in the early development of the vaccine, and we got started as fast as we could and took it as far as we could very quickly. But in parallel with that, at the beginning of 2020, having discussions to find a partner who would work with us to be the pharma partner to take on the manufacturing and all the regulatory responsibilities. AstraZeneca were prepared to work with us on that and support the university's wish that this would be made not for profit for low and middle income countries, which is really important to us. We know it's a vaccine that is suitable to use around the world, and we want it to reach as many people as possible, and we don't want it to be done at profit. The university is a charitable body, and that's really fundamentally important to all of us working on the vaccine and for the university as well. So AstraZeneca agreed to that, and we got started working together, and it was a bit of a difficult relationship to begin with because we were really busy and we had to learn how AstraZeneca work, which is obviously very different because they're a much bigger organization than our small vaccine team, and we needed to transfer all the details about the vaccine, the manufacturing process, the clinical trials, and so on. But we very quickly got into a great working relationship because we were all trying to achieve the same thing. And so our team in Oxford and the team in AstraZeneca, and then all of us working together last year, achieved such a lot because we all had a common, clear goal, which was to get this vaccine licensed, make it available on a very large scale, and that's what's happened. And so because we were all aiming at the same thing, we all just worked out what we needed to do to get there, and we got there.
Well, Dame Sarah Gilbert, thank you so much for doing this short interview. Welcome to the ISV Fellow Society. We're thrilled. We reached out to you before this happened, so yeah, I think that's really exciting that you have joined the society. And part of the society's goal is to train the next generation of scientists, and of course to lead scientific discussions of importance. And there's no one on the planet that's more important than you and your colleagues right now. So we're thrilled to have you, and we look forward to many more interactions, and congratulations on all your success.
Thank you very much. And I look forward to getting back to in-person conferences.
Yes, that will be wonderful. All right. Take care.
Thank you. Bye-bye.
Bye-bye.